Good morning. Today we are going to see about a botany. What is botany? Botany is a branch of science. It is come under biology. So the biology it is classified into two categories. One is the study of plants, that is called botany, and the study of animal is known as zoology. In this we are going to see botany chapter 1, sexual reproduction in flowering plants. So sexual reproduction in flowering plants, it is very important topic. Why? Because if there is no sexual reproduction, it is a doubt to carry out the next generation. So the sexual reproduction is carried out in flowering plant is known as angiosperms. The topic, all flowering plants will come under angiosperms, which shows sexual reproduction. So flowers are the main site for the sexual reproduction. It is colorful, beautiful and attractive, which helps in pollination. Now the first one, pre-fertilization structure and events. So several hormonal and structural changes result in differentiation and development in flower and flowering parts. So in this, the floral buds, inflorescence, they are all part of a flower. So typically a typical flower has two important structure. One is androecium, the other one is gynoecium. Androecium, the male reproductive part of a flower, and the gynoecium, the female reproductive part of a flower. These two structure plays a vital role in reproduction of flowering plants. So first we are going to see about androecium. So what is androecium? Androecium is the male reproductive part. It contains a whole of stamen. Whole in the sense the group of stamen. Their number and length are variable in different size. According to the plant's nature, according to the structure, the number of stamens are varies. Okay. First one, we are going to see the structure of a flower. So the outermost part of a flower is known as petal and the base part is known as sepal, followed by the base which carry the entire flower is known as pedicel. In this, the centermost region is covered by reproductive part of a flower. That means the male and female reproductive part of a flower. The male reproductive part is known as androecium. So in this androecium, we have stamen and anther. So the stamen is the one which contain the two main categories. One is filament, the other one is anther. So the filament which helps to attach the stamen with the pedicel, and the androecium on the top, it contains an anther which is bilobed. The anther contains number of pollen grains which helps in pollination. Now the part of a stamen, contains filament and anther. So can you see the picture? In the picture the filament and anther are clearly seen. Stamen, the basal part is known as filament and the bilobed structure is called as anther. Inside the anther it is having microsporangium. Now next one, what is the function of filament? So in the plant, the androecium part, it contains a long slender stalk like structure which is called filament. The filament which has two ends, the top end and the bottom end. So the bottom end is known as proximal end. The proximal end is attached to the thalamus. So this region is known as thalamus. The proximal end attached to the thalamus of the petal of flowers, and the top end is known as anterior end which contains anthers. If you cut open the anther, it have a transection. In this section it contains pollen grains which is tetrapod in nature, we will see later. Now the next one, anther. The anther, it is located in the topmost region of the filament. In this it contains bilobed structure. So in the filament the anther is bilobed in nature. It is bilobed. Each lobe has two thecae. This region is known as thecae. So hence it is two. It is called as dithecious. So dithecous, what is meant by dithecous? It is typically bilobed, hence it is dithecous. So often the longitudinal grooves runs lengthwise and separating the thecae. So it is bilobed, this one helps in the separation of thecae. Next one, if we cut open and see the anthers, hence anthers are bilobed, dithecous. If we cut open, the transection of anther shows tetragonal structure. That means the bilobed is having 4 structure. So left, right and below. Okay. So this 4 structure is known as tetragonal structure. Hence it is also known as tetrapods. It contains 4 microsporangia, each located at the corners. 2 in each lobe. So this one side of the lobe it contains 2 and the other side of the lobe it contains 2. The microsporangia then develop into a pollen sacs. They extend longitudinally all through the length of an anther and packed with pollen grains. So this is the detailed picture. In this picture, if we cut open the anther in a transverse section, this one is known as transverse section. If we cut open like this, it is known as longitudinal section. If we cut open into transverse section, it contains 1, 2, 3, 4. Hence it is 4. Each one is known as microsporangia. So 4 microsporangia is present in the anther. Hence the microsporangia further develop into pollen sac. Sac means bag like a structure. Inside the sac it contains pollen grains. So this is a microscopical structure of anther. Transverse section of anthers, microscopic image. In this anther sac with pollen filaments is cleanly visible in microscope. Okay. Now the next one is structure of microsporangium. So what is microsporangium? A microsporangium is present near the corner. The circle outline is known as microsporangium. Anthers have 4 microsporangium. It is surrounded by 4 wall layers. First one is epidermis. So the outermost layer is known as epidermis, followed by middle layer. In between the middle layer and the epidermis, there is another layer is known as endothecium, followed by the inside the complete round is known as tapetum. The outer 3 layers gives protection to the microsporangium, which helps in the attraction of anther and to release pollen grains. Next one, the tapetum, the innermost, the small circle is known as tapetum. The tapetum is the innermost layer which gives nourishment to the developing pollen grains. The cells of tapetum contains a cytoplasm. Cells of tapetum, the innermost layer which contain cytoplasm and nucleus. So it contains more than one nucleus. In young anther, each microsporangium has a tissue called as sporogenous tissue. I repeat it again. In young anther, each microsporangium has a tissue named as sporogenous tissue, which is located in the center. So can you see this picture? In this picture, the center region is occupied by a tissue named as sporogenous tissue. In the sporogenous tissue, it contains compactly arranged sporogenous cells. Inside that, it contains sporogenous cells. So again, the clear picture of a microsporogenesis. In this, the outer layer we already know, microsporangium. Inside that it is having a round structure which is called pollen sacs. The outer layer of pollen sacs is epidermis, followed by it is having a fibrous layer, tapetum. Inside the tapetum it is having a pollen mother cells which is diploid in nature, and pollen cells. So once the pollination occur, the cell will classified into, it will undergo pollen mother cells which is diploid in nature, which undergo meiosis. From the meiosis it divide and it become tetrad of pollen. Further it separates and it formed as pollen grains. So as the anther develops, each sporogenous cells, that is called also known as microspore or pollen mother cells. So microspores or pollen mother cells is also known as sporogenous cell, which undergoes meiotic divisions. Once it undergo meiotic division, it form microspore in tetrads. Tetrad in the sense 4. Each tetrads, that means each microspores arranged in a cluster of 4 cells. Further, the formation of microspore from the pollen mother cell through meiosis, this entire process is known as Microsporogenesis. This entire process is known as Microsporogenesis. Micro means small in structure, sporo, spores, genesis, small cells or division. So in microsporogenesis, the anthers, as the anthers mature, it dehydrate, the microspores dissociate from each other and develop into complete pollen grains. So this is the picture of matured dehisced anther. In this matured dehisced anther, it dehydrates. Dehydrate in the sense it loses its water. Once it loses its water, it started to dissociate or it started to spread up or separate. Once it started to dissociate from each other, each cell will develop into a pollen grains. Each microsporangium contains millions of pollen grains. They are released by the dehiscence of anther. It is released by the dehiscence of anthers.